Hello students, Namaskaram. Welcome to Gnana Deeksha concept-based digital learning. My name is A. Chetana, working as degree lecturer in microbiology at Telangana Social Welfare Residential Degree College, Jagathgirigutta, Hyderabad. Today, in this session, we will learn about cells and organs of the immune system. Before that, I want to give a question, that is, what are the different cells and organs used in immunity, and how these different cells involved in the immune system to provide the immunity. Structure, organization of cells and organs involved in immunity production, and how we defend ourselves with many foreign particles with the use of this immune system. Immunity, which is used to protect ourselves from the foreign particles or pathogenic organisms, it is very important in our life to fight against pathogenic microorganisms. Daily in our routine life, we are allowing many microorganisms into our body, but we are not expressing any kind of symptoms as our immune system readily attacks on these microbes and kills them. So, there is much importance and need to know about the immune system with respect to its structure and organization. Let me start the session to know the importance of the immune system with the involvement of several cells, tissues and organs. Immune system is the first line of defense. Like an army, it works to prevent the entry of microorganisms or foreign substances to inside. So, this is the first line where foreign substances cannot enter inside, is mainly by mechanical barrier that anatomically or physiologically restrict the entry of microorganisms into our body. But due to some reasons, if organism pass this barrier, escape this barrier, the second line of defense is provided by different blood cells like neutrophils, eosinophils, basophils, monocytes, macrophages, and the natural killer cells by releasing the granules from their cytoplasm. If organisms escape this second line of defense also, they should face the products produced by B cells and T cells, which are also called as B lymphocytes and T lymphocytes. Thus, immune system protects our body from foreign substances with the help of these different types of cells and organs, which we are going to see in our session. All living organisms, including plants, animals like human beings, have the capacity to protect or defend themselves from the entry of microorganisms. So, for this protection, our immune system should work like a network with a different type of cells, organs and tissues. They should work in coordinated manner to provide the protection. So, anatomical barriers where physiologically like skin, mucous membrane, cilia, stomach acid and urine flow acts as the first line of defense, as mechanically they either prevent the entry of microorganisms or trapping them. And leukocytes, which act as a second line of defense, they act by different mechanisms. Under this, lymphocytes are cells produced and matured in different lymphoid organs like primary and secondary lymphoid organs. They act as a third line of defense to neutralize the antigen nature. All leukocytes, which we generally call as WBCs, they are the main immune cells and platelets for blood clotting, and these WBCs and platelets, they reside mainly in the buffy coat, which looks like a transparent substance upon centrifugation. By adding certain chemical agent like EDTA, which acts as an anticoagulant, and this buffy coat is the layer formed between plasma and RBCs. Generally, this buffy coat is used to isolate the DNA from the blood sample and to detect parasitic infections. All the blood cells from where they have been formed. All blood cells are produced by a process called as hematopoiesis, by which RBCs, WBCs and different types of lymphocytes are produced. Normally, in a healthy human adult, approximately 10 to the power of 11 to 10 to the power of 12 new blood cells are producing each day. Hematopoiesis maintains this steady level of the cells in the peripheral circulation. Among all the different types of cells produced by hematopoiesis, the WBCs like neutrophils, basophils, eosinophils, monocytes, macrophages, and lymphocytes like T lymphocytes and B lymphocytes are mainly involved in providing the immunity to our body. By this hematopoiesis, the production starts from the hematopoietic stem cell, which acts as a multipotent stem cell, because from the same stem cell, myeloid progenitor and lymphoid progenitor are produced, and this myeloid progenitor gives to myeloid cells like neutrophils, eosinophils, basophils, monocytes, macrophages, mast cells and platelets, dendritic cells, erythrocytes, etc. And from the lymphoid progenitor, lymphocytes such as T lymphocytes, B lymphocytes and natural killer cells are produced. So, in the previous slide, we have seen the different types of cells which are producing from the hematopoiesis process. All these cells can be differentiated into granulocytes like neutrophils, eosinophils, basophils, which mainly contain the granules in their cytoplasm, and they are released out upon contact with the antigen to degrade them and to neutralize them. And all these granulocytes, they have multi-lobed nucleus. In contrast, agranulocytes such as monocytes and macrophages, they do not have any granules in their cytoplasm, so that the nucleus is very clearly visible, and these are mainly meant for the phagocytosis action. Now, we will learn these cells one after the other. First, we will discuss about the neutrophils. Neutrophils, they occupy 50 to 70%, that is the majority of the WBC population, with a multi-lobed nucleus. They are light-colored granules and stain light blue to pink with neutral stains, so that they are called as neutrophils. They have multi-lobed nucleus, so-called polymorphonuclear leukocytes. In simple term, we call it as PMNs, and the chromatin in the nucleus is very condensed. They are the first fighters against the invaded bacteria which have been entered into the body, and they are motile in nature and immediately attack the antigens and the phagocytic to engulf or eat the bacteria. Next is about eosinophils, which are also granulocytes. They stain pink or red with hematoxylin and eosin or any other acidic stain, as they have acidophilic granules in their cytoplasm, and they have two-lobed nuclei, that is bilobed nuclei, and they are also phagocytic in nature. They also engulf the bacteria, and they have important role in protection with the parasitic infections. And next is about basophils. Basophils occupy less than 1% of all the leukocytes or WBCs. The granules in their cytoplasm stain with basic stains and appear in blue color. They are non-phagocytic and plays an important role in allergies and asthma with the release of histamine granules from their cytoplasm, so that they increases the permeability of the blood vessels. And next, in contrast to granulocytes, there are also some agranulocytes among the blood cells. Among the agranulocytes, monocytes are agranular with a single lobed or bean-shaped nucleus, and they are the largest type of leukocytes which circulate in the blood for about 8 hours time period and involve in presenting the antigen which have been entered to the site of lymphocytes for an immune response, so that it acts as an antigen presenting cell. And next is about macrophages. Macrophages are nothing but they are the monocytes only, but when they enter into tissues, they become matured, called as macrophages there, and they enlarge in tissues and become phagocytic, that is engulfing the bacteria. They involve in detection, phagocytosis and destruction of the bacteria and other harmful microorganisms. You know, the macrophages are named differently based on their location, like Kupffer cells in the liver, osteal macrophages in bones, microglial cells in brain, such as etc. And they mainly carry the receptors on their surfaces for binding to an antibody. And next is about dendritic cells. Dendritic cells originate from the lymphoid progenitor from the bone marrow itself through hematopoiesis process, and they are the best antigen presenting cell, and they are also called as accessory cells. The main function is to process the antigen and present the antigen on the cell surface to the T lymphocyte or T cell. Actually, they mature themselves to present an antigen after capturing it, so that they act as messengers between the innate, that is inborn immunity, which is provided by the skin, mucous membranes, etc., and the adaptive immunity or acquired immunity, which is obtained through the lifetime. And we have seen neutrophils, macrophages, which are involved in phagocytic action. So what is phagocytosis? Phagocytosis is the process done by neutrophils, macrophages, where this phagocytic cell plasma membrane engulfs a large particle or foreign cell or an antigen, giving rise to an internal compartment called as phagosome. And then this phagosome fuses with the lysosome to form phagolysosome and mediates the destruction of the particle by forming the structures which mainly contain the hydrolytic enzymes for the degradation of the bacteria. Among different phagocytic cells, macrophages are the large cells which eat more number of the bacteria. And the next is about the lymphocytes, which are produced from the lymphocytic progenitor cell from the bone marrow itself, which acts as a stem cell by hematopoiesis, the same process. But among these lymphocytes, some of the cells migrate to the thymus, which is the primary lymphoid organ, and are called as T lymphocytes or T cells. The cells which retain in the bone marrow itself are called as B lymphocytes. So now both of the matured B and T lymphocytes migrate to the secondary lymphoid organ for interaction with the antigen and then encountering them or neutralizing them. First, we will see about the different types of lymphocytes. These lymphocytes occupy 20 to 40% of the total WBC or leukocytes. They are located mainly in the lymphatic fluid, and they are of three types: B lymphocytes, T lymphocytes, and natural killer cells, which are simply called as NK cells. So in this slide, the above picture depicts the receptor on the surfaces of these lymphocytes, which are mainly used to differentiate them. First, we will see about the B lymphocytes. They are born and originate in the bone marrow and mature there itself, or migrate to the bursa of fabricus. In case of birds, they display membrane-bound antibody on their surface. You can see the Y-shaped structure on its surface, and once it is in contact with the antigen, they get activated by the cytokines produced by the T helper cells. And then after activation, the B cells differentiates into plasma cells and memory cells. Plasma cells are involved for the production of antibodies, which provides the humoral immunity, that is where the antibodies are mainly residing in the humor or serum, and memory cells for the secondary immune response when similar antigen enters for the second time. Activation of the B cells initiates when an antigen binds to membrane-bound antibody on their surfaces. Upon their activation, they differentiates into memory and plasma cells. Here, a memory cell remembers the antigenic nature and its structure and produces secondary immune response when the same antigen enters into the body for the second time. But plasma cell produces antibodies immediately by the clonal selection, which acts on the antigen which have been entered. And the next is about T lymphocytes. They also originate in the bone marrow, but the difference is they migrate to the thymus and they activate and differentiates into T helper cells, T cytotoxic cells, and T suppressor cells. T suppressor cells are also called as T regulatory cells because they control the immune response. And T helper cells mainly produces some cytokines which are needed for the B cell activation, and they have CD4 glycoprotein receptor on their surface. These activates the B cells for their differentiation and also activates other macrophages. And T lymphocytes, mainly the TC cell, T cytotoxic cell, have CD8 receptor on their surface, and upon contact with the MHC, which stands for major histocompatibility complex, differentiates into effector cell called as cytotoxic T lymphocyte, that is called as CTL, and memory T cell. So similarly, TC cell acts both in memory cell and effector cell, and T suppressor cell or T regulatory cell have CD4 and CD25 receptors on their surface that helps in regulating or suppressing the immune system when there is no need of the immunity. T suppressor cells are also used in regulation or controlling the exaggerated immune response like autoimmune condition. And next, the third type of cells which express CD16 on their surface, which recognizes a region of the antibody, that is FC portion of the antibody molecule, are called as natural killer cells. And these NK cells are large lymphocytes compared to other lymphocytes. They are used for killing viral infected cells and tumor cells for controlling the early symptoms of the cancerous diseases. And next is about the organs of the immune system. That is the next part of our topic with the cells of the immune system. We will discuss about the organs of the immune system. So, immune system works better when both cells and organs are present in an organized manner. Immune system is organized with different types of organs which are categorized into primary and secondary lymphoid organs. Primary lymphoid organs are thymus, bone marrow, and instead of a bone marrow, bursa of fabricus is present in case of birds, where maturation of lymphocytes takes place. And secondary lymphoid organs include lymph nodes, spleen, mucus-associated lymphoid tissue, malt, gut-associated lymphoid tissue, gall, Peyer's patches of small intestine, and appendix. These are all organs of the immune system, the list of organs which provides the immunity. And next is about the thymus, that is the primary lymphoid organ. It is a bilobed organ situated above the heart region, and it is surrounded by the capsule. Thymus is divided mainly into two parts. The outer part is called as cortex and the inner part is called as a medulla. And inside of it has a network of epithelial cells, dendritic cells and macrophages. In the slide you can see clearly about its structure, and T cell maturation takes place in the thymus. T cells recognize the soluble antigen when and only presented by some antigen presenting cells like MHC, that is major histocompatibility complex. If T cells do not recognize MHC molecule, then the death of the T cells also induced by the thymus, but this thymus function decreases with the increase of the age. Initially, the epithelial cells giving rise to thymus are contagious in nature, and the cells remain connected via desmosomes between their processes, forming a sponge-like network of epithelial cells which are called as reticular epithelial cells, which is like a network. And these cells induce the lymphocytes to proliferate and distribute into the other parts of the thymus, that is the medulla and the cortex, and in this the lymphocytes differentiates into T cells, leave from there and populate the other organs. The matured T lymphocytes leave via venules into medulla and travel through the bloodstream to populate the peripheral organs. If the thymus fails to develop or thymectomy condition is seen where thymus is completely removed from the surgery, T cells do not develop, which indicates that thymus is an important organ for the T cell development. And the next primary lymphoid organ is bone marrow. which is the soft spongy tissue that is located in the medullary cavity, that is the central part of certain large sized bones. Healthy bone marrow is an essential part of the body as it contains stem cells that produce blood cells and the cells that make up the immune system. And bone marrow acts as a hematopoietic stem cell for the generation of several blood cells. B cells or B lymphocytes mature in the bone marrow itself and express the membrane-bound antibody on their surface. But in birds, instead of a bone marrow, bursa of fabricus, which is located at the cloaca region, have a similar function of the B cell maturation. And these organs, after the primary lymphoid organs, the secondary lymphoid organs are specially meant for providing the environment for the proliferation, that is a multiplication and maturation of the cells involved in adaptive or acquired immune response. These organs filter and trap the antigen that has been entered through the lymphatic fluid. Secondary lymphoid organs are lymph nodes, spleen, Peyer's patches, malt, gall and appendix. These are all secondary lymphoid organs. Among this, let me start with the lymph nodes. Lymph nodes form part of the lymphatic system which are present in most parts of the body and is connected by small lymphatic vessels. And lymph node is an encapsulated organ that is covered by a capsule with three regions, that is cortex, paracortex and medulla. Cortex contains B cells, macrophages, dendritic cells and primary follicles where B cells are present mainly. Paracortex includes T cells and dendritic cells which are antigen presenting cells, and medulla mainly contains the plasma cells which are secreting the antibodies. And the next secondary lymphoid organ is the spleen. Spleen is the organ which filters blood and traps the blood-borne antigen. It is also an encapsulated organ which is covered by a capsule and has trabeculae which divides the spleen into two portions, that is red pulp and white pulp. Red pulp mainly contains the macrophages and RBCs. Due to the presence of RBCs, they are called as red pulp. White pulp surrounding the branches of a splenic artery and forms a structure called as a pulse, that is periarteriolar lymphoid sheet. Spleen also have primary follicles which are rich in B lymphocytes. The functioning of spleen starts when blood with an antigen enters through splenic artery into the spleen. And the secondary lymphoid organ also includes the lymphatic system, which is the largest lymphatic vessel with a lymph interstitial fluid. That is, the portion of the fluid that does not enter the venous system is called as a lymphatic fluid, which is returned to the circulatory system by lymphatic vessels. And antigens are carried by this lymph to the lymph nodes. And next is about malt, which is called as a mucosa-associated lymphoid tissue. It is an organized area that surrounds digestive, respiratory and urinogenital tract regions. For example, in digestive tract such as intestine, they are called as Peyer's patches. They also include tonsils, appendix. And malt in digestive tract has a microvillus in lamina propria on their surface for absorption of foreign antigens and is absorbed to the pocket where they are engulfed by macrophages or exposed to antibodies present in the germinal centers. Peyer's patches is found throughout the ileum region of the small intestine. They check mainly the intestinal bacteria and prevent the growth of a pathogenic bacteria in the intestinal region. The function of Peyer's patches is to analyze and respond to the pathogenic microbes in the ileum region. Antigens from microbes in the gut are absorbed via endocytosis process by microfold cells lining the surface of each Peyer's patch, and these antigens are passed on to the lymphoid tissue where they are absorbed by macrophages and presented to T lymphocytes and B lymphocytes for their further action. And next is about tonsils and appendix, the other secondary lymphoid organs. Tonsils are of different types. One is lingual tonsils, which are lymphatic tissue present on surface of the base of the tongue. Pharyngeal tonsils, which are also called as adenoids, they are present at nasopharynx region. And palatine tonsils are present at the base and sides of the throat region. And these all tonsils mainly contain WBCs or leukocytes to kill the microbes and trapping them. And appendix is a blind sac, which is a vestigial sac extending from the cecum region. They hold the organism and prevent the passage of these organisms into the other parts of the intestine. Primary lymphoid organs, the main function of the primary lymphoid organs is nurturing or providing the environment for the lymphocyte development, whereas secondary lymphoid organs support the lymphocyte maturation, survival and their activation. Secondary lymphoid organs are distributed throughout the body to drain or filter the antigenic material from the tissues and the organs. For this topic, that is always health is wealth, yes, everyone agrees with this. It is provided by the immune system, which is made up of different types of cells like leukocytes, lymphocytes and primary or secondary lymphoid organs. But in turn, this strong immune system is maintained when and only we maintain healthy diet and healthy lifestyle. So, the silent army of our body, that is called as immunity, is mainly correlated to the diet what we take with good nutritional value. Simultaneously, we should also avoid stress in our life, as stress induces or activates the corticosteroid hormones, which can suppress the effectiveness of the immune system. Even all with the different types of cells, organs in well mannered, the immune system effectiveness will be lost due to this corticosteroid hormones, and even smoking, alcohol consumption also have adverse effects on all the cells of the immune system, and they influence in reducing the overall immunity of the person. We should maintain healthy diet, performing regular exercise, yoga and meditation to boost up the immune system, and immunity has to be improved to stay safe and lead a healthy life. So we summarize the cells and organs of the immune system. We have seen immunity is mainly provided by the organization of these different types of cells like leukocytes or WBCs, such as neutrophils, monocytes, macrophages, basophils, eosinophils, and lymphocytes like B cells, T cells and natural killer cells. Tissues like malt, mucus associated lymphoid tissue, GALT, that is gut associated lymphoid tissues, and organs, that is primary lymphoid organs, secondary lymphoid organs. So altogether, these cells, tissues and organs are required to maintain the immune system integrity. They are all required to provide the better immunity. Immune cells which are produced by the hematopoiesis process, they will function in different ways to fight against pathogenic microorganisms or any foreign particle or an antigen through phagocytosis, that is engulfing the bacteria, eating the bacteria, or by producing certain antibodies by plasma B cells, and or they will produce cytotoxic substances from T cytotoxic cells, which helps in cytotoxicity, either in any of the way, like antibody production by B cells, cytotoxicity by T cells, or phagocytosis by neutrophils and macrophages. The cells function in different ways, and we also have seen leukocytes, that is WBCs, act like independent, like neutrophils, macrophages, they independently act like single-celled organisms and are involved in providing the innate immunity, which is an inborn immunity that is acquired from birth itself. Lymphocytes, both B and T lymphocytes, are involved in providing the acquired or adaptive immune response, which is obtained through the lifetime when an antigen naturally enters into our body. Cells are matured. The different types of lymphocytes, they mature and prepare for encountering the antigen in both primary lymphoid organs and secondary lymphoid organs. Primary lymphoid organs are mainly used for nurturing, providing the nourishment and environment for T and B lymphocytes development, whereas secondary lymphoid organs provides the environment for multiplication and maturation of these lymphocytes. The key primary lymphoid organs of the immune system are thymus, bone marrow. In case of birds, the bone marrow function is performed by the bursa of fabricus, and secondary lymphoid organs are spleen, tonsils, lymph vessels, that is a large lymphatic system, lymph nodes which are present throughout the body, which provides a protection from an antigen, adenoids, that is tonsils located at the throat and tongue region, MALT, that is mucous associated lymphoid tissue, which is present at all the mucous epithelial cells lining the respiratory, digestive and urinogenital tract region, and GALT, that is gut associated lymphoid tissue, mainly which is present like Peyer's patches in the cecum region. So altogether the cells which are interacted with the organs, that is primary and secondary lymphoid organs, are involved in providing the immunity. So they mature at a primary lymphoid organs, they encounter the antigen at a secondary lymphoid organs. So the antigen, when it enters through different routes, either from bloodstream or from the mucous membranes or from the skin, they will be trapped by innate mechanical barriers, or if the antigen escape these innate mechanical barriers, they should face the lymphocytes which are produced from the hematopoiesis from the bone marrow. So these cells and organs of the immune system are mainly involved in providing the protection or giving the immunity to fight against different pathogenic organisms or any foreign particle. If it is entered into our body, we are not producing any type of symptoms or disease due to the presence of these different types of cells, tissues and organs present in our body. Thank you.